Assalamu alaikum warahmatullahi wabarakatuh, dear students. Today, inshallah, we're going to continue the digestive system. We're going to take the digestive glands. We're going to study the types of different salivary glands, parotid, submandibular, sublingual, and we're going to take the liver and pancreas. First, we are going to talk about the salivary glands. In order to know the salivary glands, we should know the types of acini. You know that salivary gland is an exocrine gland, contains acini and ducts. The serous acini is small in diameter, narrow lumen, lined with pyramidal cell, non-clear cell boundary, with central rounded nuclei. There is basal basophilia and apical acidophilia. While the mucous acini, large acini, lined with cuboidal cell with flat nuclei, flat basal nuclei, the cytoplasm is vacuolated due to the presence of mucus. We have another third type, which is called the mucoserous gland. I'm having a mucus acini, above it a part of the serous acini called serous demilion. Here is mucoserous gland. This is the mucus acini, vacuolated cell with basal flat nuclei, and there is the serous demilion. Again, mucus acini, above it the serous demilion or the crescent of Gaianzi. Mucoserous gland. The duct system, we have inside the lobule ducts known as intralobular duct. We have the intercalated duct and we have the intralobular duct or the striated duct. Here is the acini and here is the duct inside the lobule. You can see here the duct is showing basal acidophilic striation. Here is the acini, basal basophilia and apical acidophilia. Here, dear students, is the parotid gland, and you know that the parotid gland is purely serous gland. Only serous acini, contain only serous acini. As we said before, inside the lobule, I'm having very small duct known as intercalated duct and striated or intralobular duct. Between the lobules, we have interlobular duct. Between the lobes, we have interlobar duct, and then we have the main duct. Again, all of the acini are serous acini, and I'm having intralobular ducts. Here, the fat cell present in the trabeculae known as adipocyte. This is a lobule, and this is another lobule. Excuse me. And this is connective tissue contain interlobar duct. From where I know it is interlobar, look at the lining epithelium. It appears pseudostratified columnar epithelium. The intralobular is cuboidal. The interlobular is columnar. The interlobar is pseudostratified, and then the main duct stratified columnar, then stratified squamous and open, and the duct is called stencil duct, opens against the upper second molar tooth. So it is purely serous acini, and I can see the ducts. Inside the lobule, intralobular duct is called striated duct. Between the lobes, here is the interlobar duct, pseudostratified columnar epithelium. Dear students, here you can see serous acini and mucoserous acini. Many serous acini and some mucoserous acini. So it is called submandibular gland. Submandibular gland of human, mainly serous acini and some mucoserous acini. Here, dear students, is the sublingual glands. I do not have pure serous. I'm having mucus and mucoserous. Mucus and mucoserous acini. This is intralobular duct. Again, dear students, this is the parotid gland. Why? All the acini are serous acini. all the acini are serous acini. This is human submandibular gland. Why? I can see many serous acini and some mucoserous acini. Here is human sublingual. I'm having only mucus and mucoserous, no pure serous acini. No pure serous acini. Here is the pancreas. Here is the pancreas. The acini is serous acini, as that of the parotid, but the basal basophilia and apical acidophilia is so evident, and here the nuclei is basal, not central. And I can see here something in the lumen of the acini, which is the nucleus of the intercalated duct. As the first duct arises from the pancreas is called intercalated, and it is invaginated inside the acini. While I'm doing cut section, I can see the nucleus of the intercalated duct at the lumen of the acini. As you can see here, dear students, the acini, basal basophilia, apical acidophilia, and I can see the intercalated duct, which is called invaginated inside the lumen, called central acinus. Again, dear students, you can see here the serous acini. You can see the serous acini, basal basophilia and apical acidophilia. Here is the lumen of the duct here present in the pancreas, lined by simple cuboidal epithelium. The endocrine portion of the pancreas is known as islets of Langerhans, aggregates of cells, beta, alpha, delta, and other cells, surrounded with fenestrated blood capillaries. Here is the acini, intercalated duct lined with simple squamous epithelium, and here is the islets of Langerhans. The last gland we're going to take is the liver. You know, the liver is again mixed gland, compound tubular, partially covered with peritoneum, formed of stroma and parenchyma. The stroma is formed of capsule, trabeculae, and reticular connective tissue. The functional unit and the structural unit of the liver known as classic hepatic lobule. Another classification in the parenchyma of the liver, here is the classic hepatic lobule with central vein at the center. Here something called portal lobule, in triangular in shape, in which the apex of the triangle is central vein of 3 adjacent classic hepatic lobule, while the liver acinus between 2 adjacent classic hepatic lobule, drawing a diamond shape. Here the best blood supply here in this zone A, then less than the least around the central vein, which is known as zone 3. Classic hepatic lobule, hexagonal, it is the functional and the structural unit, drawn by trabeculae, which is not very well demarcated, present mainly in liver cirrhosis or in liver pig and camel. In human, it is very faint, as you can see. Here at the corner, there is portal tract containing branch of the portal vein, branch of hepatic artery and bile duct, as well as lymph vessel. In the center, we have the central vein. Higher magnification. This is the central vein radiating from it the hepatocyte. The hepatocyte are acidophilic, vacuolated as it is rich in glycogen. Some cell contain 2 nuclei because it's very active. These cells arranged in rows. Between the rows, I cannot see the bile canaliculi. Between the plates, I can see spaces which is known as blood sinusoids. This is central vein, this is hepatocyte, and these are the sinusoids. These sinusoid lined with endothelium and von Kuffer cell. This is the central vein. These are the hepatocyte. Any space you can see in the data show is the blood sinusoid. Canaliculi cannot be seen by the light, seen only by EM. So here the blood sinusoid lined with endothelium and von Kuffer cell. What the von Kuffer cell? It is a phagocytic cell. Any phagocytic cell has monocyte origin. The cell is irregular. Cell membrane is irregular to do the phagocytosis during pseudopodia, rich in lysosomes, the lysosome produced by the Golgi apparatus and the rough endoplasmic reticulum. How to stain any phagocytic cell? By something called vital stain. I'm going to inject the stain while the animal alive. 3 to 4 hours, I'm going to sacrifice the animal. You're going to see stained cells. These stained cells are the phagocytic cell that stain this stain. So it's called vital stain. Example, ink and ink or turban blue. Again, the portal tract. Here, portal vein, branch of the portal vein, thin wall, wide lumen, branch of hepatic artery, thick wall, narrow lumen, and bile duct lined by simple cubical epithelium and small lymph vessel. Branch of the portal vein, branch of hepatic artery and bile duct. These are the hepatocyte. As I told you, any space is the blood sinusoid. The gallbladder here, it is related to the liver. It is folded, lined with simple columnar epithelium. As you can see here, it's not very well developed smooth muscle, not in layers. Okay. Again, highly folded mucosa, irregular smooth muscle, highly folded mucosa and irregular smooth muscle. Now we're going to check our knowledge by the data show exam. What this type of acini? It's mucus acini. Why? Large, lined with large cuboidal cell with flat basal nuclei, clear cell boundary, and the cytoplasm is vacuolated. This is serous acini. Why? Small, narrow lumen, basal, basophilia. The nucleus is little bit central to basal, and here is the intralobular duct. So A, mucus acini, B is serous acini. Here, dear students, look at A. This vacuolated thing is the adipocyte present in the trabeculae. All the acini is serous acini, small, narrow, deep basophilic cytoplasm. Here, so A is parotid. B, I'm having serous acini and some mucoserous acini. So it is submandibular gland of human. Here I'm having no pure serous acini, mucus and mucoserous. So it is human sublingual gland. Here, dear students, I'm having serous acini. Small, narrow, basal basophilia, and I'm having mucoserous. So, as I have serous acini here, so it is not. Why it is not parotid? Because I'm have mucoserous. Why it is not sublingual? Because I'm have pure serous acini. So I'm having serous acini and mucoserous. So this gland is submandibular gland of human. Submandibular gland of human. SA serous acini, M mucus, SD serous demilium. Okay, dear students, what is this? I can see serous acini with very well formed basal basophilia and apical acidophilia. In between them, I'm having cells, which is the islets of Langerhans. So this is pancreas. I can see islets of Langerhans A, B, the serous acini, pancreatic acini. This is section in the liver, the parenchyma of the liver. The black shape is classic hepatic lobule. The green shape is the portal lobule, while the red shape is liver acinus. Here, dear students, is a section in the liver. I can see here the classic hepatic lobule demarcated by the trabeculae, and this is central vein. Okay, so the structure is classic hepatic lobule, the star is the central vein, and the blue arrow are the trabeculae. Or the sept, trabeculae or sept. Dear students, what this section is, it's liver. The black arrow points to any space is the sinusoid. The yellow arrow points to the hepatocyte. Usually, dear students, prepare question visible feature of identification from where you know that it is hepatocyte. It appears acidophilic, polyhedral cell, acidophilic, cytoplasm, pale nucleus with prominent nucleoli. Then the arrowhead points to the central vein. Here, dear students, is a section in the gallbladder from where you know folded mucosa, and the lining epithelium is simple columnar, and the star is the muscle layer, irregularly arranged smooth muscle layer. Here, dear students, the label diagrams. This is parotid gland. Why? One, only serous acini. Only serous acini. Small, narrow lumen, cuboidal cells, pyramidal cells, non-clear cell boundaries. The nucleus is central. Inside the lobule, I'm having intralobular duct. Intralobular duct. Here is the trabeculae. Between the lobules, I'm having interlobular duct lined with simple columnar epithelium. Between the lobes, I'm having interlobar lined with pseudostratified columnar epithelium. So how could I differentiate between the intralobular and the interlobar? Look at the lining epithelium. It appears as if more than one, so it is interlobar duct. Pancreas, I'm having serous acini, basal basophilia, apical acidophilia. And I'm having the islets of Langerhans. The beta cell present at the center appears basophilic, small and large number. The alpha cell appears at the periphery, large in size and small in number, and these are defenestrated blood capillaries. Something lined with simple columnar, it is the interlobar. The interlobar duct. The last photo is for the liver. I can see classic hepatic lobule, central vein radiating from it, hepatocyte. In between the rows, there is bile canaliculi, but I cannot see them. Between the plates, I'm having blood sinusoid. At the corners, I'm having the portal tract, which is formed of branch of hepatic artery, portal vein, bile duct, and lymph vessel. Thank you, dear students. We have finished the digestive glands of module 206. Assalamu alaikum wa rahmatullahi wa barakatuh.